Good morning, my dear students. Today I am going to tell you about 3 questions. The first question is nucleus, second question is bacterium, and third question is viruses. The first question, nucleus. Consider what is nucleus. Nucleus is an essential organelle typical for eukaryotes. Nucleus functions: storage of genetic information. Second function, the regulation of all vital processes in the cell. There are 4 parts of nucleus: nuclear envelope, nucleoplasm, liquid content of nucleus, chromatin, and nucleoli. On this slide you see electron micrograph of nucleus. You can see nuclear envelope, but you cannot see that nuclear envelope consists of 2 membranes. Don't see on this micrograph. Dark parts, it's heterochromatin, a light parts, nucleoplasm with euchromatin. In the center of nucleus you can see small dark point nucleoli. Function of each part consider later. So you can see on this electron micrograph mitochondria, a lot of cavities of endoplasmic reticulum, granular endoplasmic reticulum, because you can see ribosome on the surface. You studied the rough endoplasmic reticulum always surrounded nucleus. Nuclear envelope. What you see nuclear envelope on this slide more detailedly. You see 2 membranes of nuclear envelope, outer membrane and inner membrane. Nuclear envelope is formed by 2 membranes. Outer membrane is often a continuation of the granular endoplasmic reticulum and bears on the surface of the ribosome. Inner membrane smooth. It enters into a complex with the internal structure called lamina. Lamina involved in maintaining the shape of the nucleus, also in the packing of chromatin. Lamina consists of intermediate microfilaments. On this slide, you see surface of nuclear envelopes. And this point, it's a lot of nuclear pores on the surface. Nuclear pores. Nuclear envelope contains numerous nuclear pores. Each pore consists of a ring of proteins, you see, with a central channel, the whole complex being secured to and stabilized by the nuclear lamina. Nuclear pores regulate the extent of metabolites, macromolecules, and ribosomal subunits between nucleus and cytoplasm. And you see outer membrane of nuclear envelope, inner membrane contained with lamina, nuclear lamina, which consists of intermediate microfilaments, and complex of nuclear pores from a lot of protein. Ring of protein from 8 proteins. Chromatin. Chromatin is a complex of DNA with histone proteins. There are 9 of histone proteins for period 8-A, 8-B, 8-3, 8-4, and 1 unperiod 8-1. 8 proteins form a globular structure, octamer, and you see octamer from 8 histone protein, period 8-A, 8-B, 8-3, and 8-4. Together 8 proteins. Around each octamer, almost twice, the DNA helix is wound, forming a nucleosome. То есть octamer with DNA is called nucleosome. DNA between nucleosome enters the complex with the 9th protein histone 8-1. This DNA is called linked DNA. 8-1 take part to the further DNA packaging, and this level of DNA packaging is called a nucleosomal level. On this slide you see nucleosome which consists of octamer and DNA. On the next slide, consider more detailedly level of packaging of DNA. There are euchromatin and heterochromatin. Euchromatin is almost not visible in the light microscope and contains functionally active DNA. From this DNA can read information. Heterochromatin has the form of grains and clumps in the light microscope, contains functionally inactive DNA. Reading of information does not occur from this DNA. On this slide you see the first level of packaging of DNA, nucleosomal level. You can see DNA molecule with surround octamer, and octamer with nucleosome is called, with DNA is called nucleosome, nucleosomal level. After that, nucleosomes join together and form next structure, solenoid structure or a chromatin fiber. The diameter of this structure 30 nanometer. So you can see next level of packaging of DNA. The first level, DNA double helix forms a filament diameter of 2 nanometer, as you see on this slide. Short radius of DNA double helix, 2 nanometer diameter. This DNA is screwed onto octamer and forms nucleosomes. Nucleosomes are part of the nucleosomal filament with a diameter of 11 nanometer. This structure is often called beads on a string. This level of packaging of DNA, it's euchromatin, where DNA can be active. Next level, nucleosomal filament is twisted and forms a chromatin fiber with a diameter of 30 nanometer. After that, chromatin fiber forms looped domains with a diameter of 300 nanometer. More tightly packed loop domains form a condensed section of chromosome with a diameter on 700 nanometer. They are part of a metaphase chromosome having a diameter of 1400 nanometer. То есть you can see nucleosomal level of chromatin fiber, level with looped domains, and formation of metaphase chromosome. And repeat this material. You see on this slide DNA double helix. At the simplest level, chromatin is a double-stranded helical structure of DNA. Then DNA is complex with histone, form, and nucleosome. Each nucleosome consists of 8 histone proteins around which the DNA wraps 1.65 times. A chromatosome consists of nucleosome plus 81 histone. Yellow on this slide. The nucleosome folds up to produce a fiber, chromatin fiber, with diameter of 30 nanometer. That forms loops averaging 300 nanometer in length. The 300 nanometer fibers are compressed and folded to produce a 250 nanometer wide fiber. And tight coiling of the 250 nanometer fiber produced the chromatid of a chromosome. A common diameter of chromosome 1400 nanometer. Nucleolus. In the center of nucleus, you can see nucleolus. What is nucleolus? Nucleolus is an electron-dense body, is a place where accumulate a large number of organic materials, copies of RNA, ribosomal proteins, also large and small subunits of ribosome around the fragment of DNA encoding RNA. The function of the nucleolus is the formation of ribosomes. Nucleolus are heterogeneous structures. There are the paler areas being the sites of DNA which code RNA, and the dark areas being the place where of partially assembled ribosomes. The many different ribosomal proteins are imported from the cytoplasm and conjugated with the ribosomal RNA to form the ribosomal subunits, which then pass into the cytoplasm before being assembled into fully active ribosomes. You must remember the main function of nucleolus formation of subunits of ribosome, because nucleolus have fragment of DNA with encoding RNA. A ribosome consists of RNA and proteins. Second question of our lecture, bacteria. Bacteria are single-celled organisms. They are prokaryotes because no nucleus. The bacteria cells have no nucleus. The circular double-stranded DNA is free located in the cytoplasm. And you see on this slide. Bacterial DNA forms a complex with the non-histone protein forming a nucleoid. So in the center of bacteria, you can see nucleoid. It's electron micrograph of bacteria and you can see nucleoid in the center. Nucleoid consists of DNA, double-stranded circle DNA of bacteria, and non-histone proteins. Bacteria cells surrounded by an outer membrane. The cell wall is arranged outside the membrane and comprises a polysaccharide murein. Some bacteria may be curved with a mucous capsule. On this slide you can see three structures of envelope of bacteria. Inner structure, plasma membrane. Outside, cell wall from urine, plasma membrane and cell wall. То есть all bacteria have plasma membrane and cell wall from urine, but only some bacteria has mucous capsule on the surface. On this picture you can see double-stranded DNA in the center, or a bacterial chromosome or a nucleoid. Liquid contains cytoplasm, a lot of ribosome, purple point in the cytoplasm. Invagination of plasma membrane is called mesosome, and maybe additional DNA is called plasmid. And you can see flagella, it may be a lot of pili on the surface of bacteria. Next picture about bacteria. Also you see nucleoid radion, nucleoid, each double-stranded circle DNA with non-histone proteins. Cytoplasm, blue. Black point, each ribosome. Food granule or inclusions in cytoplasm. Additional DNA plasmid. A lot of pili. It folded of plasma membrane. Maybe prokaryotic flagellum. Structure of prokaryotic flagellum different from structure of eukaryotic flagellum. Because bacterium does not have microtubule and microfilaments. No element cytoskeleton in cytoplasm. And you see three structure of bacteria envelope: plasma membrane, cell wall, and mucous capsule. And a new picture of bacteria. Nucleoid, inclusions, small points, its ribosome, additional DNA plasmid, flagellum, pili, and three structure of bacterial envelope. On practical class, you must name these elements of structure of bacteria. This will be 1, 2, 3, and you must say what is 1, what is 3, what is 4, and other. The shapes of bacteria. Prokaryotic cells have various shapes. The four basic shapes of bacteria. Cocci is spherical shape, bacilli rod shape, and you see cocci on this slide. Maybe coccus, maybe Diplococcus, when two cocci together. Diplococci encapsulated, maybe streptococci chain from cocci, sarcina, tetrad, staphylococci. Bacilli or a rod-shaped. Maybe bacilli, diplobacilli, streptobacilli. Polycytes. Spirocheta, spiral shape. Typical spirocheta you see on this picture. And vibrio, coma shape. Typical vibrio you see on this picture. And other intermediate shapes between one or second types. And different shapes of bacteria you can see on this slide. Coccus, Diplococcus, Bacillus, Spirilla, Vibrio. Nutrition of bacteria. By way of nutrition, bacteria can be divided into autotrophic and heterotrophic organisms. Autotrophic bacteria are divided into photosynthetic bacteria and chemosynthetic bacteria. Two types. Heterotrophic bacteria are divided into parasites, saprotrophs, and symbionts. photosynthetic autotrophic bacteria. You see example on this slide. You see photosynthetic cell, for example, Anabena, this bacteria. And there are different types of cell. Photosynthetic cell and heterocyst, vegetative cell, photosynthetic, primary metabolic function, photosynthesis. Heterocyst, nitrogen fixation function. And akinet, dormant spore forming cell. Chemosynthetic bacteria use chemical reactions between inorganic substances to generate energy, which they use to synthesize organic substances. On this slide, you see type of chemosynthetic bacteria. Hydrogen bacteria use this reaction to generate energy to synthesize organic substances. Colorless sulfur bacteria, iron bacteria, nitrite, nitrite bacteria. And you see reaction with typical for sulfur bacteria. Carbon dioxide, water, hydrogen sulfide, and result formation of sugar and sulfur compounds. Heterotrophic bacteria. There are three types. The first type, bacteria parasites. These bacteria feed on living organisms and cause various diseases. For example, pneumococcus causes pneumonia, you see on this slide. Vibrio cholerae is a cause of cholera. Helicobacter pylori can lead to the development of stomach ulcers. Bacteria saprotrophs. They feed on the dead remains. These bacteria convert the organic matter of the dead bodies in minerals, thus closes the cycle of substances in nature. And third group of heterotrophic bacteria, bacteria symbionts. These bacteria form a mutually beneficial relationship with other organisms. For example, some bacteria in the human colon helps to digest cellulose, creates an acidic environment where harmful bacteria and fungi do not develop, are involved in the formation of some vitamins. For example, vitamins of group B and K. On this slide, you see lactobacillus under the microscope. It's bacteria symbionts in our colon. So we considered new types of nutrition of bacteria. You must remember two types, autotrophic and heterotrophic. Autotrophic divide on chemosynthetic and photosynthetic bacterium. Heterotrophic three group, bacteria saprotrophs, bacteria parasites, and bacteria symbionts. Bacteria can be divided into aerobic and anaerobic bacteria. An example of anaerobic bacteria is bacteria that live in the human intestine. An example of aerobic bacteria is putrefaction bacteria, which decompose dead organism along the circulation of substances in nature. The main features of bacteria. The bacteria survive in the form of spores in adverse conditions and for a long time can survive, maybe more 100 years. Reproduction of bacteria. Bacteria reproduce through asexual reproduction, usually by binary fission. A process of asexual reproduction you can see on this slide. What occurs? Double-stranded circle DNA bacteria replicated. So firstly, cell elongated and DNA is replicated, and you see two sets of DNA. After that, cell wall and plasma membrane begin divide. Cross-wall forms completely around divided DNA. And cells separate, and you can see two new bacteria. Each asexual reproduction. Sexual reproduction. Bacteria do not have a sexual reproduction, but there is sexual recombination. There are three types of sexual recombination of bacteria: conjugation, transformation, and transduction. Process of conjugation, it's very difficult, and I want to show you short film from YouTube about this process, but name of author of this film I don't know, but you always find it in YouTube if need.
Bacterial conjugation is a process of genetic transfer between bacterial cells that requires direct contact between the cells. Many, but not all, species of bacteria can conjugate. Conjugation can occur between cells of the same species or even between cells of two different species. A small DNA circle or plasmid called the F factor is required for conjugation. The F factor stands for fertility factor. Strains of bacteria containing the F factor are called F+. Those without it are called F-. An F+ cell, or donor, produces a structure called a pilus to connect with another recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a protein assembly called the relaxosome, which associates with the strand to be transferred, or the tDNA strand. Accessory proteins of the relaxosome are released, but a portion of the relaxosome called the relaxase remains attached to the tDNA. This tDNA relaxase complex is recognized by a coupling factor and transferred to the exporter, a complex in the F+ cell that is contiguous with the pilus. The exporter pumps the tDNA relaxase complex into the recipient cell. Once the entire tDNA molecule is transferred to the recipient cell, relaxase joins the ends to make a circular DNA molecule. As the tDNA is transferred to the recipient cell, it is replicated to become double-stranded. In the donor cell, the F-factor DNA was also replicated to become double-stranded. This actually occurred as the tDNA was being transferred to the recipient cell. In the end, both cells wind up with a complete double-stranded copy of the F-factor. Their connection through the pilus is released, and each is now an F+ cell that can go on to conjugate with other cells.
So, conjugation. Some bacteria, in addition to the basic DNA, has additional DNA called plasmids. Donor bacterium having the plasmid may form cytoplasmic bridges by using sexual rod with a bacterium which has no plasmid. First, double-stranded plasmid DNA is separated into two chains. One of the chains is transferred to the recipient bacterium. Then each bacterium completes the missing chain on the principle of complementarity. And you see this process on these pictures. You see plasmid, which consists of two chains, process formation of bridges between two bacterium. Bacterium which has plasmid is called F+ bacterium. Bacterium without plasmid is called F- bacterium. Donor bacterium and recipient bacterium. Donor cell attached to a recipient cell with its pilus. Formation bridge between two bacterium. The pilus draws the cells together. The cells contact one another. One strand of plasmid DNA transfers to the recipient, and after that the recipient synthesizes a complementary strand to become an F+ cell. The donor synthesizes a complementary strand, restoring its complete plasmid. And as a result, you can see two bacterium which have plasmid. Each process of conjugation. Next process, transformation. Transformation is transfer of a fragment of DNA from dead bacterium to living bacterium, where the fragment replaces the homologous site. Thus, a gene that provides resistance to antibiotic can pass from a dead bacterium, donor cell, to a living bacterium, recipient cell, that did not previously have this property. And next type, third type of sexual recombination, transduction. Transduction is transfer of DNA fragments from one bacterium to another by using viruses. This viruses is called bacteriophages. Penetrating into the bacteria cell, the virus multiples and forms a protein capsid. Sometimes virus capsid randomly encompasses fragment of bacterial DNA. This forms a transductive particle. When such a virus infects a new bacterium, it introduces into it not the viral DNA, but a fragment of the DNA of the first bacterium. And the second bacterium acquires new properties that the first bacterium had. And about transduction, I want to show short film from YouTube.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the GAL gene, near the specific site of insertion remains attached to the phage DNA and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome, but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
So you must remember 3 types of sexual recombination: conjugation, transformation, and transduction. And next question today, viruses. Viruses are organisms having no cellular structure. They consist of a nucleic acid, DNA or RNA, which is surrounded by a protein shell called a capsid. Above the capsid may be located a lipoprotein envelope, which often is a fragment of the membrane of the host cell from which the virus was released. And you see on this slide virus of influenza and bacteriophage. The difficult structure around DNA is protein capsid. Bacteriophage has head, tail, fibers, difficult protein capsid. And virus of influenza has capsid, white line, and additional envelope above the capsid, a lipoprotein envelope, which often is a fragment of the membrane of the host cell, and a lot of receptor on the surface. Metabolism, growth, and development are processes that are not typical for viruses. Viruses don't have metabolism, growth, and development. The protein envelope capsid of the virus consists of separate unit capsomeres. Capsomeres can form capsid of various shapes and symmetries. There are viruses with helical capsid symmetry, for example, virus tobacco mosaic, and a cubic capsid symmetry, for example, herpes viruses. And on this slide, you see a lot of shapes of viruses: tobacco mosaic viruses, adenoviruses, influenza viruses, and bacteriophage. Picture and electronic micrograph. Viruses are permanent parasites that exhibit properties of living within the host cell. Heredity, variation, and reproduction are typical for viruses. Outside the host cell, viruses behave like structure of inanimate nature. Therefore, viruses is a bridge between the animate and inanimate nature. Viruses are very specific. They penetrate only into the cell of a specific type. Viruses have the receptor. You see a lot of receptor on this slide. Viruses have the receptor to the specific cells. For example, hepatitis B virus affects the liver cells, the human immunodeficiency virus affects blood lymphocytes. On this slide, you can see human immunodeficiency virus. You see 2 chains of RNA, special enzymes, reverse transcriptase, capsid, with red or a purple color, and additional envelope, lipoproteins, an envelope with a lot of receptor in the surface. The behavior of virus in the cell. The virus recognizes the cell by using receptors. It then enters the cell by one of the methods. Animal viruses enter the cell by phagocytosis, and you see this process on the slide. The receptor of viruses join with a receptor on the surface of the cell, and process phagocytosis occurs, and viruses penetrate into the cell. Plant viruses and fungi viruses penetrate into the cell through the damaged cell wall. Viruses, bacteria, bacteriophages, enter the cell by injection. So, the main state of the life of the virus. The first state, absorptions of viron on the cell with help receptor. Next state, penetration of viruses into the cell by phagocytosis or through damaged cell wall or by injections. Next process, deproteinization of the virus, the release of the virus from its envelopes. Next state, synthesis of viral components, replication of viral nucleic acid, and synthesis of viral protein that forms the capsid. After that, self-assembly occurs. The viral capsid captures and surrounds the viral nucleic acid. And formation on viruses and their exit from the cell. And you see this stage on this slide. This first process, adsorption of virus on the cell with help receptor. After that, penetration of virus into the cell by phagocytosis on this slide. Then deproteinization occurs. Lysosome of host cell join with phagosome which contain viruses within, and enzymes of endosome digest envelope of viruses. And you see DNA or RNA of viruses in the cytoplasm of host cell without protein or a lipoprotein envelope. After that, replication of DNA or multiples RNA occurs, and you can see a lot of copies of DNA, virus DNA in cytoplasm of host cell. And after that, with help organelles of host cell, synthesis of viral protein occurs and form a lot of capsid. Next stage, self-assembly, capsid. Capsid captures and surrounds the viral nucleic acid and form new viruses. After that, virus exit from the cell on this slide by exocytosis. Use plasma membrane of host cell and use receptor, specified receptor. And you see progeny virus virion. The AIDS virus contains two strands of RNA and enzyme called reverse transcriptase. But you see this virus penetrate into the cell by phagocytosis too. First, a DNA strand complementary to the viral RNA is formed by reverse transcriptase enzyme. То есть, firstly, deproteinization occurs, and you see RNA, viral RNA, and after that, with help special enzyme, transcriptase enzyme, DNA strand form on the base virus RNA. Next stage, the DNA strand then forms complementary second DNA strand. Thus, a double-stranded DNA fragment is formed. It travels to the nucleus, where it is incorporated into the host cell DNA molecules. So this fragment of DNA incorporate into the host cell's DNA molecule and function as a DIN. After the virus is activated, it forms a viral RNA and viral RNA multiples, and on the base viral RNA, a lot of protein of capsid of virus forms. Self-assembly takes place and new viral particles are formed. And viral exit from cell by exocytosis. And this process you see on this slide too. Virus penetrate host cell, nuclear envelope of host cell. After that, replication occurs and formation of virus envelope capsid. After that, self-assembly occurs and form new viruses. Same process on this slide. Significance of viruses, the value of viruses. Viruses regulate the number of other living organisms. Virus is element of biosphere. Viruses cause diseases in animals and plants. Scientists use viruses to create vaccines against diseases. Genetics use viruses as vector to move genes. For example, in agriculture, viruses is a biological method of controlling plant pests. So, today we considered 3 questions. Nucleus, structure of nucleus. Remember, nucleus consists of 4 parts: nuclear envelope, nucleoplasm, chromatin, ochromatin and heterochromatin, and nucleolus. Second question, bacteria. And third question, viruses. On this week, the topic our class work will be nucleus. So you must remember structure of nucleus for practical class on this week. And level of packaging of DNA. What is nucleosome? What is chromatin fiber? And structure of nucleus. Today our lecture is over. Thank you for attention. See you later. Goodbye.